Everyone, my name is Robert Iestwaart. I'm a postdoctoral fellow in the lab of Sterling Churchman at Harvard Medical School. Thank you to the ISCB for organizing and Seth for hosting today the Posterpalooza. I'm very excited to share with you my recently published work on GeneWalk. So, as you all know, high throughput experiments generate big data. So a very typical experimental setup is to have a control versus treatment experiment, and followed by like a next generation sequencing assay like RNA-seq or NAT-seq or CRISPR screens to get a phenotypic readout. And often what you then end up with is over 1,000 differentially expressed genes that change their expression in the treatment versus the control. And then the question becomes like how to follow up on this long list of genes of interest. So I've developed a new algorithm tool called GeneWalk, which was published like just over a month ago. So I hope you're excited to try it out after hearing a bit more during this talk. So before I dive into GeneWalk, I think a common follow-up approach that has been out there and has been developed over the last 30 years is co-enrichment analysis, which really gives a global relevance of annotated gene sets. So if you have a list of differentially expressed genes, you perform the analysis, and what you'll get is like these, these significant go annotations like chromatin organizations, which correspond to annotated gene sets, which are known to involved in those processes. So this is very interesting. It's very important because it's fast, it's unbiased, and it gives you context-specific information, right? It tells you what's happening in the experiment that you're performing. But it's not gene specific. It's kind of specific gene set level. So many people have developed this over the last 20 years. And so what I'd like to introduce is GeneWalk, which is context specific for individual genes of interest. So if you have like your favorite differentially expressed gene X, we'll list the go annotations, and what GeneWalk will provide then is like a relevance score for each individual go annotations, so that the context-specific gene functions for this gene X are then the go annotation with a high relevance score. In this short talk, I'll briefly outline the methodology, and then we'll apply to a benchmark ground case study. So GeneWalk takes up a list of genes of interest, like for instance your differentially expressed genes from the experiment, and then it will automatically assemble a context-specific gene network, and that only contains the input genes and all the known relations that are known in the literature between them. And then GeneWalk adds the go ontology over here, appends this network to the gene regulatory network and connects the go terms with the genes through their go annotations. Then we perform network representation learning to convert each node in the network to vectors, so that we can do significance testing for a particular gene of interest like MYC. It can then calculate the cosine similarity and rank each go term according to their relevance. So the context-specific gene functions are then the go annotations with high similarity significance. So due to the time, I don't have too much. I don't have time to go into this aspect, but let me talk a little bit more about the network representation learning algorithm. So once we have the network, we can sample random walks to get a large set of training data. Basically for a neural net, but it basically gets a large set of neighboring node pairs. And these neighboring node pairs, the frequency of them occurring really reflects the network topology. So we can then train this neural network, basically a word of a single hidden layer, where the input node is basically one of these nodes, and then the output is the corresponding neighboring node pair, so that based on the frequency of how often these occur, and the the network learns basically the vector representations. So the vector representations are then for each input node like these the kind of the hidden layer weights. Okay, and this this has been developed. It's called GeneWalk, this algorithm, and it's been developed for social media networks. And we, the novelties, we apply it to gene regulatory networks, like with our particular network, to kind of apply to this specific function of gene function relevance scoring. And then the second part that I developed was basically a statistical test for these cosine similarity significance. So what we do here is we randomize networks so that we get random vector representations.
Sorry, everyone. Sorry,
Yes.
Robert. You got disconnected.
Okay. Sorry.
When you went to the last slide.
So then
you need to share your screen again.
Okay. Share my screen again. Yeah. Sorry.
That's quite all right. I don't think this one was your fault.
Yes. Let me try again. Yeah. All right. So I think you see my screen now.
Yep, there we are.
All right.
Perfect.
Sorry for the
right when you got
interruption.
to this slide that it dropped out on us. Sorry about that.
Okay. Yeah. So the statistical, what I added beyond the GeneWalk vector network representation learning is also a statistical test to get the significance for the gene functions. So how does this work? So we have the GeneWalk network. We randomize the network. Sorry, let me add my pointer. So we randomize this network whilst keeping the node degree distribution the same, and then we can again apply the GeneWalk algorithm to get vector representations, but now not only for the GeneWalk network but also for the randomized network. And using these randomized vector representations, we can generate a null distribution of cosine similarities between the random nodes, which basically provide a null distribution for the real connectivity. So when we compare this cosine similarity between a gene and a Go term, we can then compare it to the null to get a p-value, and then we can do multiple testing correction for multiple gene annotations. So at the end of the day, the context-specific gene functions are then the Go annotations with a significance cosine similarity. Yeah. So let's try to apply this to one benchmark case study. So here we look at a mouse quaking gene, QKI, and this gene, when it's deleted conditionally in oligodendrocytes, then the myelin sheath, which normally cover the neuron axons to increase the speed of signal transmission, are basically degraded. So this is like a very well characterized phenotype, and the readout that we're looking at is RNA sequencing, and this has been published previously in a publication. So we looked at this transcriptional data, the RNA-seq, when we did differential expression, and one of the strongest downregulated genes is MAL, which stands for myelin and lymphocyte protein. And this gene has really been characterized to be involved in this myelination. And what does GeneWalk predict? Indeed, like the structural constituents of myelin sheath and myelination are top ranked, while the other terms are not significant. So this is kind of, for at least for this gene, giving insight into, correctly predicting what the function of that gene is in this particular experiment. But can we do this more generally? So, and can we compare how GeneWalk does compared to these global enrichment methods? So the enriched the alternative methods to basically give a ranked list of go terms, so that we can, for each gene, like MEL, just take significant terms and rank them by their global relevance score. And then we can compare them to a ground truth, like myelination relevant, but other terms not relevant. So what we do is, when we do this for Panther global enrichment analysis method, this would be the ranking according to Panther. So myelination is up there, but protein binding, which is a very generic term, is even more strongly enriched, so it ends up at the top. Well, structural constituent myelin sheath, even though it's relevant for MEL, right? We know that it's actually in this myelin sheath, and that it's, you know, that this process is affected by the knockout. It actually not globally enriched, so it ends up at the bottom. So this is kind of indicating how GeneWalk is different from global enrichment methods. And so we basically did this systematically, comparing with 8 other methods. And what we did is we now have we extended this benchmark beyond MEL to all systematic to a systematic benchmark where that we created based on the published literature. So we basically looked up all the genes that were listed in that paper and all the functions that were mentioned in the paper related to those genes, so that we get a systematic benchmark and then compare each method. We see that GeneWalk kind of does best in this ranking task, which means, like, are the relevant functions ranked on top versus the functions that are not the co-annotations that are not mentioned in the paper are ranked near the bottom. And also for a binary classification task, is this function relevant or not? Like, is it mentioned in the paper or not? Then GeneWalk also again does better. So yeah, as I said, we have recently published this work. We also have made a tutorial, and we've made our code on GitHub online to really try to make it easy for people to run. And we hope that this will make people excited to try it out on their own data. So before I go to the acknowledgement slide, I just want to spend one slide on a shout out on one other previous paper that I published last year, which is Machine Learning Guided Association of Adverse Drug Reactions with in vitro target-based pharmacology. So here we basically used in vitro pharmacology on over 2,000 drugs to kind of see what gene targets what protein targets these drugs bind to. And we can connect this with observed adverse drug reaction reports for these drugs, and then use random forest to learn the associations between these targets and the adverse drug reactions, so that we get more insight into which genes might actually be causing these adverse drug reactions. So with this, we found over 221 gene target ADR as statistical associations. So I hope that will be interesting for future research to investigate more the mechanistic basics of adverse drug reactions. So with that, I'd like to acknowledge my mentor, Sterling Churchman, lab members from the Churchman Lab, and our collaborators from the Sorga Lab who helped with the network assembly part, and also the John and Ben on the software implementation of GeneWalk. So with that, I'd like to thank you for your attention, and happy to take